Good morning. Let's start chapter 10, and that is your senses. We're going to start with some basic information. Remember, we sense everything with our brain. So when you feel something on your finger is not the one that feels that sensation. Your brain is the one that perceives the information. So we have to make sure we understand that this information is going to go from peripheral to our brain or spinal cord. So first we have to change stimuli into action potential. That is called transduction. That means changing the stimulation into nerve impulse, to action potential. You hear the sound, you feel something, all this information from the sound, from the light, from the pressure, from touch, from the heat or temperature needs to be transduced into action potential. Also, you have to understand that every nerve delivers a specific impulse. If that nerve carries the light information, brain understand that must be for vision. Based on the type of signal that your receptor can transduce, we have different type of receptors. We have chemoreceptors that they transduce and they understand chemicals in the case of taste or smell. We have photoreceptors that they can detect and transduce the sense of light. We have thermoreceptors that they can transduce and receive the information from change of temperature. And we have mechanoreceptors. Obviously, they are going to transduce the information that comes to them based on mechanical deformation. Example for this one would be touch and hearing. We're going to talk about that one also. In this table of your book, you can see there are different type of receptors. It tells you what type of a stimulation these receptors can detect. And here's some examples. You can take a look at those and you can summarize the information by the end of this chapter. Now let's talk about the most important receptor, and that is, of course, nociceptor. Nociceptor is a receptor for pain. This receptor can detect damage to the tissue. A damaged tissue can release some neurotransmitter, which is mainly can be glutamate or substance P. These two can stimulate nociceptor, and this receptor is going to send the information to the brain. We already know that to block this pain, we're going to release something that's called endogenous opioid. I think it's worth to be mentioned that not only substance P and glutamate can stimulate nociceptors and create the impulse, but also other stimulation like heat, cold, or pressure also can stimulate nociceptor. When the heat, cold, or pressure is beyond normal limit. At the beginning, heat is going to stimulate your thermoreceptors. At the beginning, cold is going to stimulate your thermoreceptor. At the beginning, pressure is going to stimulate your mechanoreceptor. But if heat becomes so hot, if the cold becomes so cold, and if the pressure becomes so high, then they can stimulate the nociceptor. So at the normal range, these three, they can stimulate their own receptors. But if the heat or cold or pressure goes beyond normal, they can also stimulate nociceptors. We have two different type of neurons that carries pain. Some of them that are myelinated, and the other one that are unmyelinated. The myelinated, as you know, is going to carry the information much faster than unmyelinated. Therefore, for the sharp and sudden pain, we use myelinated neuron, and for dull pain, we use unmyelinated neuron. We mentioned that nociceptor can be stimulated by damage to the tissue. You can also stimulate this pain receptor by ATP or change of pH. We have different type of receptors in different parts of the body. There are some receptors that are found inside the skin. Those are receptors that are going to detect touch and pressure and heat and cold and pain. There are some receptors that they are found in the muscle or tendon or joints. Those receptors are called proprioceptors. Proprioceptors are receptors that detect position of the joint and the status of your tendon. For instance, if you close your eye and if I bend your knee, you know that your knee is bent. If I grab your big toe and move your big toe, you know that your big toe is moving because you have a sense of proprioception. Proprioception is a sense that your body knows exactly where your joints are. When there's any change in the joint, when any change in the length of the tendon, the sense of proprioception is going to get activated and you know that your joint is moved. And finally, we have some receptors. They are not in the skin and they are not in the muscle, but they are called spatial senses: vision, hearing, taste, smell, and equilibrium. Receptors can react toward stimulation in two different ways. Some receptors are called phasic receptor. A phasic receptor is a receptor that adapts. So at the beginning, it's going to send stimulation, but if the stimulation continues, then the receptor stops sending the information. Example, smell. Someone is going to come to the room and is going to wear cologne. As soon as it's going to come inside the room, everybody is going to smell that cologne. But after a while, you don't smell it anymore because your receptor is going to get adapted. When you are wearing your watch, at the beginning you're going to feel it, and after a while you don't feel it because the sense of touch can be adapted. I mean, the same token, when you walk inside a cold room, at the beginning you're going to feel it, but after a while you're going to adapt to that temperature. Other receptors are not adaptable. They are tonic receptors. In other words, as long as the stimulus is there, they're going to send information, like pain. You don't feel pain at the beginning and then the pain is going to go away. Pain is going to be there as long as the stimulus is there. This picture shows the phasic receptor and it shows also the tonic receptor. As you can see, the stimulation continues, but here it's going to fade out. Another topic that I like to talk about is called spinal gating. So what's spinal gating? Well, let me explain that one by using this picture. When you feel something painful, this information from, let's say in this case, from your finger is going to go to your spinal cord and then it's going to go to your cortex, and that's how you're going to feel it. If all this information goes from your finger to your brain is going to go nuts. We have to decrease this stimulation before it's going to get to the brain. What you feel in your brain is a fraction of what really happens if you don't have this spinal gating. So what's spinal gating? Is a process that decreases the pain before reach to the brain. So this information is going to get subsided, it's going to get alleviated before even brain notice it. And how does it work? The answer is everything is going to start from the midbrain. Midbrain, as you know, is part of your brain stem. Midbrain has an area, it's called periaqueductal gray region. If you don't know what it is, just learn that is an area inside the midbrain. And that area is called periaqueductal gray region. From this area, there's some neuron that is going to come down to the spinal cord. This neuron that comes from this area to spinal cord, they have a name. They are called reticulospinal tract. From midbrain to spinal cord, obviously we have to pass through medulla, and that's exactly what's going to happen. The neuron first is going to come to medulla, it's going to synapse in the medulla, and it's going to come to the spinal cord. In the spinal cord, this neuron is going to first release serotonin. This purple one is the serotonin. The serotonin is going to activate this interneuron. Remember the term interneuron. Interneuron is a small neuron that is found inside the spinal cord or is found inside the brain. Look at this neuron. This neuron is in the spinal cord, but part of it is inside the brain. So this is not interneuron. Interneuron by definition is the whole length of the neuron is in one structure. The whole length of this neuron is inside the spinal cord. So that's why it's called interneuron. So this neuron is going to come to the spinal cord and release serotonin. And it's going to activate this interneuron. This interneuron at the end is going to release enkephalin, as you know, is an endogenous opium. And this endogenous opium is going to block this neuron. You can see this neuron is taking information from spinal cord and trying to get it to the brain. By blocking this one, so brain is going to receive less painful stimulation. Remember, this neuron is going to bring information to spinal cord and attach to second neuron. So this is second neuron, this is first neuron. In the pathway I just showed you, this interneuron is going to block the second neuron. That is called a postsynaptic inhibition. Postsynaptic, because after the synapse. We can do this inhibition by presynaptic inhibition, before the synapse. This one is going to release also enkephalin. So enkephalin can do postsynaptic inhibition or can do presynaptic inhibition. And as a result, the amount of pain that reach to the cerebral cortex will diminish. This is called spinal gating because you can close the gate on the spinal cord and make sure less information is going to get to your brain. This pathway that takes the pain to the brain is called spinal thalamic tract because it's going to go from the spinal cord first to the thalamus and then it's going to go to the cortex. But this one that calms down is called reticulospinal tract. Let's move on to the spatial senses. Among the spatial senses, of course, the vision and hearing would be the most important one. So we're going to go through the taste and the smell fairly quickly to get to the hearing and then vision, and we're going to discuss that one a bit more in detail. So let's start first with the taste. Another name for taste is called gustation. In your tongue, you have some structures called taste bud. Taste buds are the receptors for detecting taste. Taste bud is inside the structure that we call it papilla. Here is the tongue. Here's the surface of the tongue. And you can see there are some tiny bulges we call papilla. Papilla that they contain taste bud is circumvallate, is this one, and also is a foliate, and also is fungiform. Filiform papilla does not have taste bud. So there are three papilla that has taste bud. One is circumvallate, which is toward the back of the tongue, foliate toward the side of the tongue, and fungiform, which is on the anterior part of the tongue. If you look at one of those taste bud, you can see your taste bud is composed of some cells. It's called receptor cells that they're going to detect the taste. Then there are some supporting cells. We have basal cells. And these receptor cells, they have some microvilli that they're going to open to the surface of the tongue. Here's the surface of the tongue. This hole that connects this taste cell to the surface of the tongue is called taste pore. So when you eat food, this particle of the food through this taste pore can reach and connect with this taste receptor cell. Taste receptor cell is going to detect this chemical, is going to transduce it, and is going to create action potential, and is going to send it to your brain. Let's start with circumvallate. Circumvallate, we said, is in the posterior surface, and they are the largest. I'm sure you noticed this one. Circumvallate is the largest. As far as the number, there are not that many, but as far as the size, they are huge. They have so many taste buds. In fact, almost half of the total taste bud is inside the circumvallate papilla. Circumvallate papilla sends information to the brain through cranial nerve 7 and 9. Mostly 9. If that's a taste bud inside the circumvallate, they're going to grab the information and they're going to send it to the brain. And this nerve that takes this information to the brain is cranial nerve 7 and 9. Mostly, of course, 9. We said foliate is the one that is on the side of the tongue. They're going to send the information about the taste through cranial nerve 9, or it's called glossopharyngeal. And finally, fungiform papilla is the one that is found in the surface of the tongue. They are more concentrated at the tip of the tongue, and they're going to get the information and send it to the brain through cranial nerve 7. And you know, cranial nerve 7 also is called facial nerve. So these taste buds, based on where they are, they grab the information and they're going to send it through the cranial nerve either 7 or 9. And first it's going to go to medulla. From medulla it's going to go to thalamus. And from thalamus it's going to get to your brain. Remember, everything must go through thalamus except the sense of smell. Every sense that goes through the cortex of the brain is going to pass through thalamus except the sense of smell. From thalamus, we're going to get to insula. Remember, insula is a primary gustatory cortex. And then from there, you can also send this information to parietal lobe and prefrontal cortex. You will see that the reason you send it to this area because the sense of smell and sense of taste, they share the information together. And that's why when you get cold, you don't smell or you don't taste food. Now, we have 5 different types of taste. In this picture, you can see the taste of bitter, sweet, and umami. Here shows one of these taste buds, one epithelial cells. By the way, the taste bud that receives the information and detects the information is a modified epithelial cell. Let's say in the case of bitter, usually the molecule is quinine, is going to sit on the receptor and is going to stimulate a G protein. This G protein is going to create a second messenger. This second messenger is IP3 and calcium. We talked about different type of second messenger briefly. We know that IP3 and calcium are second messengers. So for bitter, this G protein is going to create IP3 and calcium. And as a result, it's going to open up a channel. It's called TRP. TRP stands for transient receptor potential. By opening this channel, you're going to release a neurotransmitter that stimulates this nerve. This nerve can be cranial nerve 7 or can be cranial nerve 9. I put the sweet and umami also because they use the same pattern. Although the G protein is different, it's not the same G protein, but that G protein also is going to increase IP3 and calcium. and also opens the TRP channels. Bitter, sweet, and umami, all three senses, they're going to use G protein, although different type of G protein, but at the end is going to increase IP3 and calcium. In the case of sour, molecule is H. H is going to get inside the cell directly. Imagine that if that's H, it's going to go through and it's going to come in. So there is no G protein. H is going to enter the cell. When H is going to enter the cell, H is going to block the potassium channel. And you know, by blocking potassium channel, the cell becomes depolarized. In the case of salty, of course, the main component would be sodium. Sodium is going to get inside the cell through, of course, sodium channel and depolarize the cell, stimulate the nerve ending. And that is going to bring us to the end of the sense of taste. Let's move on to the sense of smell. This is inside the nose. The odor is going to come inside the nose. In the roof of your nose, we have some neurons that are hanging inside. So here, this part, this is this bone, it's called ethmoid bone, and here are the axons that are hanging inside your nose. These neurons that detect the odor are embedded in the olfactory epithelium. So odor is going to come, and this cilia at the end of this neuron is going to detect those chemicals. The information is going to go up, it's going to pass through this bone, and it's going to enter to your brain. Inside the brain is going to join another type of cells. It's called mitral cells. These are mitral cells. The mitral cells and the olfactory neurons synapse in the area is called glomeruli. So these are glomeruli. These are mitral cell. Mitral cell is going to get together and create olfactory tract, and it's going to get information to a part of the brain for perception of the smell. It's called piriform cortex. It's part of the temporal lobe, but also it's going to send it to amygdala. Remember, amygdala was a fear center. Some animals, they can smell their predators, and as a result, they run away. In human, of course, is as part of evolution, is not as important, but they also make some connection with frontal lobe and parietal lobe. In this pathway, they also make some connection with hippocampus, which we already know is part of the limbic system. Remember, we said smell is going to change your mood as part of the limbic system. When your mood changes, your learning ability is going to change. Remember, frontal lobe also received the sense of taste. So that's why taste and smell in the frontal lobe, they connected each other. That's why I just mentioned that when you get cold, then you cannot taste food either. The main olfactory cortex would be in the temporal lobe, in a place that we call it piriform cortex. This picture is going to summarize what you saw over there. Here, inside the nose, the odor is going to come. Here's the cilia of the neuron that can detect it. The neuron is going to bring this information and is going to pass through the ethmoid bone. Part of the ethmoid bone is called cribriform plate that has holes on it. This neuron is going to pass through these holes. It's going to make a synapse with mitral cells in the area we call glomerulus. This part of the sense of smell that is inside the nose is called olfactory epithelium, and the other side in the brain is called olfactory bulb. Sense of smell, just like taste, is a chemical sense. There is a chemical reaction here. We have over 380 different genes that create over 1,000 different receptors for us. Of course, different people, they have different ability to smell different odors, but in general, we have 380 genes and we create 1,000 different type of receptors, protein. But now, how does the smell works? Let's take a look at this diagram. The odorant, the chemical, is going to sit on the receptor, and this receptor is attached to the G protein. This G protein, again, is a different G protein. It's called GOLF for olfaction. And this G protein is going to break up, and alpha unit is going to move and is going to activate adenylyl cyclase, and that is going to become active, breaks down ATP to cyclic AMP. Cyclic AMP is going to come and open the channel that was closed before, and the result of this one would be entrance of sodium and calcium. So therefore, your neuron becomes stimulated. So the answer that how do we smell things? The answer is using G protein and a channel that is sodium and calcium channel. And this is going to bring us to the sense of ear and hearing.